all types of RNAs that are formed. What types of RNAs? Types of RNAs. Three types of RNA: ribosomal RNA, messenger RNA, transfer RNA. All these three types of RNA that are produced from DNA by transcription. And all these three involved in protein synthesis. Ribosomal RNA provides space and also enzyme, act as the enzyme during the protein synthesis. mRNA that carry message from the DNA that which type of protein to be synthesized. tRNA that translate the codons present on the mRNA and also brings the amino acids to the protein synthetic areas. So the three different types of RNAs that are involved in the three different functions during protein synthesis, all these three are essential for protein synthesis. All these three types of RNA that are formed from the DNA by this method, transcription, they perform different functions. Here the two types of organisms, prokaryotes and eukaryotes. And all the three types of RNA that produced by polymerase. What is that polymerase? DNA dependent RNA polymerase. Enzyme involved in the formation of all types of RNAs. DNA dependent RNA polymerase. DNA dependent RNA polymerase is essential for the formation of any type of RNA. In prokaryotes, types of DNA dependent RNA polymerases, only one type. Only one type of RNA polymerase that synthesizes all types of RNAs in case of the prokaryotes. But in eukaryotes, in nucleus, three different types of RNA polymerase is present. In cytoplasm, that means in cell organelles, normal RNA polymerase is present. That means total types of RNA polymerases present in eukaryotes. Eukaryotes. Total types of RNA polymerases present in nucleus. Three types. Cell organelles, one type. Total 4 types. Types of RNA polymerases present in eukaryotes, 4 types. 3 types of RNA polymerases present in nucleus. 1 type of RNA present in cell organelles. That is mitochondria, chloroplast. These are the cell organelles, show protein synthesis. Now, come to this in nucleus. What are the 3 types of RNAs, RNA polymerases, and what function they perform? Polymerase, RNA polymerase 1. Produce ribosomal RNA. Ribosomal RNA. RNA polymerase 1 is involved in the synthesis of ribosomal RNA. Here, ribosomal RNA 28S, 18S, RNA polymerase 1 that synthesizes ribosomal RNA. This ribosomal RNA, that is the 3 types, 28S ribosomal RNA, 18S ribosomal RNA, 5' ribosomal RNA. Next, RNA polymerase 2, that is involved in the synthesis of precursor of mRNA, that is heterogeneous nuclear RNA. Heterogeneous nuclear RNA is the precursor of mRNA. mRNA finally formed from the heterogeneous nuclear RNA. RNA polymerase 3, that is involved in formation of transfer RNA, small nuclear RNA, small nuclear RNA, and ribosomal RNA, a small fragment of ribosomal RNA. So, RNA polymerases involved in formation of ribosomal RNA, polymerase 1 and polymerase 3, tRNA synthesis polymerase 3, small nucleolar RNAs polymerase 3, hnRNA polymerase 2, only ribosomal RNA polymerase 1. So, in prokaryotes only one type of RNA polymerase, but in the eukaryotes 3 types of RNA polymerases. They are involved in mainly RNA polymerase mainly involved in the synthesis of the ribosomal RNA. RNA polymerase 2 mainly involved in the formation of the mRNA. Of course, that is formed from the hnRNA. And RNA polymerase 3 that is involved mainly in the formation of transfer RNA.